Prokaryotes form two of the three major life forms on Earth. They're nearly all multicellular, and they are all prokaryotic, meaning that they lack a membrane-bound nucleus. While both bacteria and archaea are almost completely unicellular, there are certain multicellular species. In order to be considered multicellular, they have to have intercellular connective structures linking those cells together. These can form strings or mats known as colonies. However, in a colony, there is no differentiation of cells, in which distinct tissues are produced, which is common in most multicellular eukaryotic organisms. Here we have an example on the right of a multicellular bacteria in which cells are differentiated, and what you see are spores, which are fruiting bodies of the bacteria. Bacteria and archaea are everywhere and have been on Earth for a really long time. They have been found in every ecosystem from 40 miles above the Earth's surface in the atmosphere to the bottom of the ocean, from the boiling pools of Yellowstone to underneath glaciers of the North Pole. They're inside us, all over us, in the air we breathe, and all over the food that we eat. At 3.5 billion years old, they are the oldest living organisms known. In fact, bacteria and archaea had the Earth all to themselves for over half of their existence. Scientists have spent the several past centuries cataloging the species of the world and have come up with a current estimate of 1.8 billion species of plants and animals, and it's estimated to be around 9 million. In contrast, only 5,000 species of bacteria and archaea have been cataloged. This isn't because that there aren't any, it's because that they're really hard to identify. With today's technology, we will not even be able to be close to estimate the number of species that exist, let alone the number of individual prokaryotes that live on this Earth. However, that didn't stop a group of microbiologists from the University of Georgia from trying. They estimate the number of individual bacteria to be 5 million trillion. That's a 5 with 30 zeros after it. If you look at it this way, if each bacterium were a penny, the stack would reach 1 trillion light years away. These almost incomprehensible numbers give only a sketch of the vast pervasiveness of bacteria in our natural world. Did you remember to floss this morning? Well, as it turns out, it is a continuing battle. In fact, an adult has 10 to the 14th human cells within its body. But in your digestive tract alone has an order of magnitude more bacterial cells. An order of magnitude! We have a little less living on our skin, only 10 to the 12th power. So when someone says they really aren't feeling like themselves today, ask them if they think bacteria might be trying to take control of their body, because it might be true. Prokaryotes are everywhere. They live in the outer reaches of the atmosphere, inside our stomachs and boiling water, underneath glaciers of the North Pole, at the bottom of the deepest part of the ocean, and even miles below the Earth's surface. Even though they are super tiny, they are thought to make up over 10% of the world's biomass. Here is a true color picture of the Grand Prismatic Springs of Yellowstone National Park. It really is that color. This is not photoshopped. It is a geological feature that constantly oozes water superheated from the Earth's interior. As the water oozes out, it flows into extremely shallow streams. All those colors you see, well, other than blue, are unique bacterial communities that thrive at different temperatures: lemon yellow, mustard yellow, light orange, dark orange, and even red. And there isn't a plant in sight. Bacteria are the primary agents of disease. Bacteria that cause disease are said to be pathogenic, and they primarily contaminate tissues at entry points such as mouths, eyes, and open wounds. And in plants, bacteria can also cause disease, and they are also most susceptible to infection at entry points, such as stomata, the breathing pores of leaves, and any open wounds the plants may have. Periodontitis is a disease of the mouth that primarily causes gum decay, and it is the first discovered Archaean pathogen. Periodontitis involves progressive loss of bone around the teeth, and if left untreated, can lead to the loosening and subsequent loss of teeth. With that said, there are very few pathogenic archaea that affect human health. The germ theory of disease is a theory that proposes that microorganisms are the cause of many diseases. Although highly controversial when first proposed, germ theory was validated in the late 19th century and is now a fundamental part of modern medicine and clinical microbiology, leading to such important innovations as antibiotic and hygienic practices. Robert Koch was the first scientist to devise a series of tests used to assess the germ theory of disease. Koch's postulates were published in 1890 and derived from his work demonstrating that anthrax was caused by the bacterium Bacillus anthracis. These postulates are still used today to help determine whether a newly discovered disease is caused by a microorganism. Koch's postulates are 4 criteria designed to establish a causal relationship between the causative microbe and a disease. The postulates were formed by Robert Koch and Friedrich Loeffler in 1884 and refined and published by Koch in 1890. These postulates have been generalized to many other diseases. These postulates are the following. The microorganism must be found in abundance in all organisms suffering from the disease, but should not be found in healthy organisms. The second one, the microorganism must be isolated from a diseased organism and grown in a pure culture. Third, the cultured microorganism should cause disease when introduced into a healthy organism. And lastly, the microorganism must be re-isolated from the inoculated, diseased experimental host and identified as being identical to the original specific causative agent. Once microbiologists brought attention to the fact that germs create disease, many cities around the world began instituting cleaning campaigns. And you can see the effect of this. The mortality rate has consistently dropped since the beginning of the 20th century. Everyone thinks antibiotics are what dramatically reduced mortality rates. However, penicillin, which was the first antibiotic that was widely distributed, wasn't widely used until the late 1940s. By this time, the mortality rate had been cut from approximately 800 per 100,000 to 150 per 100,000. So sanitation was really the key component in controlling pathogenic prevalence. Antibiotics were the nail in the coffin, however, but they were not without their consequences. The ability for bacteria to resist disease is known as its virulence, and virulence varies among individuals of a population, which is why there is such concern about the overuse of antibiotics. Within a given population, it is known that certain individuals are going to be resistant to antibiotics. Subsequently, those individuals will reproduce and produce the next generation. This is a picture of E. coli, and interestingly, its virulence depends on its genome. Discovered in 1928, antibiotics were a game changer in modern medicine. In fact, they are what defines modern medicine. By World War II, the use of antibiotics were so commonplace that they were given out to young children as preventative medicine, much like we do vaccines today. However, this has led to an unfortunate conclusion. Simple natural selection theory proposes that those individuals most fit to survive in a population do and reproduce, providing the seed for the next generation. What we have seen is that bacteria have become resistant to antibiotics because of the normal evolutionary processes. This has led to antibiotic strains of bacteria that are scary to think about the implications of. We live in an extreme world. The X Games were created to celebrate that. We do 1080, kick-flip McNasties, double flips off of bicycles, surf in hurricanes, and ski 89-degree slopes. However, bacteria and archaea are the original extremophiles. They can literally grow on salt crystals where nothing else can. They can live and thrive in temperatures barely below boiling to temperatures barely above freezing. And they can exist in extremely high pressure environments, even 7 miles below the surface of the ocean. Wow! These prokaryotes that grow in extreme environments are really important to biological sciences. They are the weirdest organisms that we know, and their ancestry is so old that looking at them may help explain how life on Earth came to be. It is likely that life began in the absence of sunlight. In these environments, geothermal energy is the physical source of energy that is used to generate chemical source for life to carry out its functions. These organisms are so weird that they are best guess as to what to look for on other planets for extraterrestrial life. In the movies, we always see extraterrestrial life that looks an awful like us: bipedal, big forward looking eyes, and even a mouth and a nose. However, the most likely organisms that we'll actually find will look something probably more like prokaryotes. I could be wrong, but if I were a betting man, I'd bet on finding little boogers before finding little green men. Researching extremophiles has tremendously advanced our knowledge of history of life on Earth, and not just because it allowed us to see that there are truly 3 domains of life on Earth, not 5 kingdoms. Researchers looking at the bacterial mats in Yellowstone found a protein that allowed researchers to amplify segments of DNA of any organism, which allows us to sequence those organisms. Without this technology, genetic research would likely still be in its infancy, simply because we would not be able to see the A, C, T's, and G's that we need to compare taxa. So how do we find a new bacterial species? Well, it isn't nearly as easy as finding a new insect species. If you want to find a new insect species, just go someplace not a lot of entomologists have been, like North Korea, or someplace where there's a huge amount of biodiversity, like Brazil. Take your butterfly net and start frolicking around the bushes. You're very likely to come up with a new species. And how do you know it's a new species? You just look at it and ask yourself, self? Does that look like a new species? If so, keep it and take it back to the lab for further testing. Oh, and just for the record, most entomologists choose sipping margaritas in sunny tropical Brazil over the work encampments of North Korea. So to find a new bacterial species, we have to do the above steps. But we have to do even more in the lab. We have to sample cells from a specific environment. The most studied environment is the human body, for obvious reasons. Of the 5,000 species we know of, 10% live in the human mouth. Just think of all the other environments where bacteria live, and you might go cross-eyed. So the cells are collected, but that's just the beginning. The cells are taken back to the lab where they have to be enriched. They must be grown, usually in a petri dish, into a colony, simply so there'll be enough of them to actually study. And here's the tricky part. Nearly all bacteria have to be grown under very specific environmental conditions. If you get one of these wrong, the bacteria won't grow. Once you grow a colony up that's large enough, you have to sequence part of its genome. In this way, nearly all bacterial species that have been identified using the phonetic approach under the phylogenetic species concept. So how do you sequence a gene? It is really expensive and very time-consuming to sequence an entire genome. So researchers have identified specific genes in which they isolate from the rest of the genome in order to compare it with other species. That isolated part of the genome is then amplified to increase the abundance of it. Then the amplified genes are put into a sequence machine that can identify the sequence of nucleotides of the DNA, the A, C, T's, and G's. Then researchers can compare the new sequence to see if their sequence significantly differs from others. If it does, then it can be argued that you have discovered a new species. However, how significant does the DNA have to be in order to consider it a different species? This is extremely subjective and a hot point of contention amongst microbiologists. Until the 20th century, most biologists considered all living things to be classifiable either as a plant or an animal. But in the 1950s and 1960s, most biologists came to the realization that this system failed to accommodate the fungi, protists, and bacteria. By the 1970s, a system of 5 kingdoms had come to be accepted as a model by which all living things could be classified. At a more fundamental level, a distinction was made between the prokaryotic bacteria and the 4 eukaryotic kingdoms: plants, animal, fungi, and protist. The distinction recognized the common traits that eukaryotic organisms share, such as a nuclei, cytoskeletons, and internal membranes. The scientific community was understandably shocked in the late 1970s by the discovery of a new group of organisms, the Archaea. Carl Woese and his colleagues at the University of Illinois were studying the relationships among prokaryotes using DNA sequences, and they found that there were 2 distinctly different groups. Those bacteria that lived at high temperatures or produced methane clustered as a group well away from the usual bacteria and the eukaryotes. Because of this vast difference in genetic makeup, Woese proposed that life be divided into 3 domains: eukaryota, eubacteria, and archaeabacteria. He later decided that the term Archaea bacteria was a misnomer and shortened it to Archaea. He also showed that bacteria were the oldest group of organisms on Earth today. And he also showed that Archaea and eukaryotes were more closely related than bacteria are to Archaea. That's pretty interesting. Prokaryotes are really diverse. They can vary in size from a half a micron to 100 microns. That would be like if humans varied from 5 centimeters to 10 meters. 10 meters is like 3 stories tall. Their shapes also have 3 major variations. They can look like rods, spheres, or even rotini pasta. And the way they get around can vary from whip-like flagella, which allows certain bacteria to sort of swim, and others have a gliding motion, reaching out to a destination and pulling into it, much like certain worms. And others have hairs on their membranes called cilia that make them move around with the efficiency of a hovercraft. Some prokaryotes have a thicker cell wall that contains a lot of carbohydrates, and when it's stained, appears dark purple in color. These are said to be gram stain positive. Other prokaryotes lack an abundance of carbohydrates and contain an extra phospholipid layer and don't absorb gram stain. They're called gram stain negative. Organisms that make their own food are said to be autotrophs. Let's break down that word. Auto means oneself, and troph means to nourish. So autotrophs nourish themselves. We know and love autotrophs every day. We know them as plants. Well, certain bacteria can produce their own carbon-containing compounds, usually sugars, as well and are also known as autotrophs. Organisms that rely on other organisms to produce carbon-containing compounds and consume those organisms for food are known as heterotrophs. Hetero means different and troph means to nourish. We are heterotrophs, deer are heterotrophs, and bacteria that don't produce their own food are also heterotrophs. Remember cellular respiration and photosynthesis? In photosynthesis, plants utilize energy from the sun and combine carbon dioxide with water to make sugars and oxygen gas. Plants break down those sugars using oxygen as an electron receptor in order to synthesize ATP from ADP in a phosphorus molecule, in a process known as cellular respiration. This is responsible for generating the energy needed for plants to go through life's functions. Animals also break down sugars in exactly the same process. Certain prokaryotes also have these metabolic processes. However, the 6 known metabolic processes in living organisms, prokaryotes do them all, where eukaryotes only do 2. So plants, animals, fungi, and anything else you can really think of only really do these 2. Prokaryotes can use another electron donor other than sugar, and they can use another electron receptor other than oxygen to go through cellular respiration. The production of ATP using energy of the sunlight is called photophosphorylation. Only 2 sources of energy are available to living organisms: sunlight and reduction oxidation reactions, otherwise known as redox reactions. All organisms produce ATP, which is the universal currency of life. In photophosphorylation, light energy is used to create a high-energy electron donor and a lower-energy electron acceptor. Electrons then move spontaneously from donor to acceptor through an electron transport chain. An electron transport chain couples electrons transfer between an electron donor, such as NADH, and an electron acceptor, such as oxygen gas, with the transfer of hydrogen ions, or you can think of them as protons, across a membrane. The resulting electrochemical proton gradient is used to generate chemical energy in the form of ATP. Electron transport chains are the cellular mechanisms used for extracting energy from sunlight in photosynthesis and also from redox reactions such as the oxidation of sugars in the form of respiration. Chemoorganotrophs are organisms which oxidize the chemical bonds in organic compounds containing carbon, as their energy source. Chemoorganotrophs also attain the carbon molecules that they need for cellular function from these organic compounds. The organic compounds they oxidize include sugars, like glucose, as well as fats and proteins. All animals are chemoheterotrophs, meaning they oxidize chemical compounds as a source of energy and carbon, as are fungi, protozoa, and some bacteria. The important differentiation among this group is that chemoorganotrophs oxidize only organic compounds, while chemolithotrophs instead use inorganic compounds as a source of energy. A chemolithotroph is an organism that uses an inorganic substrate, usually of mineral origin, to obtain reducing equivalents for use in biosynthesis, for example, carbon dioxide fixation, or energy conservation via aerobic or anaerobic respiration. Known chemolithotrophs are exclusively microbes. No known macrofauna possess the ability to utilize inorganic compounds as energy sources. Macrofauna and lithotrophs can form symbiotic relationships, in which case the lithotrophs are usually called prokaryotic symbionts. An example of this is the chemolithotrophic bacteria in deep sea worms, in which the bacterial lithotrophs use the worm's body as a host, but they produce ATP from the deep sea vents and share the energy with their hosts, living in perfect symbiosis.